All right, welcome everybody to the NMDC SFA webinar. My name is Emily Elo Fedrosh, and I am delighted to have this webinar to share our work on the NMDC program. In terms of logistics, we have a shared Google Doc that will be used for the Q&A session. Certainly you are welcome to use the Zoom Q&A link if you have immediate questions, but in case you are not able to access the Google Doc, please use the Q&A. I would like to also mention that this webinar will be recorded and posted to share with others who are not able to attend today. And I'll let you know in a few minutes that when we start the recording, we will turn the recording off for the Q&A session, and then we'll go through some logistics for the Q&A once we get there. All right, so let's get started. I am now, let's see. I am now starting the recording. Is the recording on?
Yeah. Yeah.
Oh, perfect. All right, great. So welcome everybody. Here's an overview for what we'll be covering today during this webinar. We are joined by Ramana Madupu, our DOE Program Manager for the NMDC, as well as many other DOE Program Managers on the call. I will be providing an overview of the NMDC, our vision and our mission for this pilot program. And then I'll be joined by David Hayes, Sam Purvine, Bin Hu, Kirsten Fagnan, Pajav Vangay, and Alicia Wood-Charlson to go through some of the logistics and an overview of the various activities and resources that we offer as part of the NMDC. And then we will be discussing some ways that you can partner with the NMDC as part of your SFA research activities. So at this point, I would like to hand it over to Ramana.
Thank you, Emily. Good morning, good afternoon to all. As a prelude to this meeting, I wanted to call to attention Bssd's goals for the computational biology portfolio. The computational program is part of the larger genomic science effort and is very much enabling capability within Bssd. So in keeping with BER's strategic plans, Bssd has been making sustained investments to support development of open access cyber infrastructure platforms for systems biology research. For example, the KBase, the DOE Systems Biology Knowledge Base KBase, and bioinformatics capabilities within JGI. So the availability of high throughput multi-omics techniques is revolutionizing biological research. The generation of omics data types, which is genomic, transcriptomic, proteomic, and metabolomic, and other relevant data types, has become more or less routine to investigate microbial ecosystems and key environmental processes. So Bssd is committed to support development of integrative open access computational platforms to combine data sets with analytical applications to be able to accelerate collaborative and multidisciplinary science. So as part of this larger goal to develop and sustain community resources, Bssd funded the NMDC program, the National Microbiome Data Collaborator, which you will hear about more today, in FY19 for a period of 3 years. So the vision, as Emily stated, the vision for NMDC is to make microbiome data fair and also to provide easy access to harmonized and standardized microbiome data sets to the community to facilitate data exploration, discovery, and also provide a way of collaboration and provide access to integrative data science ecosystem. So today, we will hear from the NMDC team about how NMDC can support integration of microbiome data across multiple studies and data types, and how the community of researchers can engage with NMDC to deposit their data sets as well as access its various functionalities. Next slide.
All right, great. Thanks so much, Ramana. And so we're really delighted to have this opportunity to pilot the NMDC program over the next several months. As Ramana mentioned, we've been in operation since July of 2019, and we've had a lot of different activities over the course of that time to develop capabilities that link and coordinate across many of the existing DOE resources, and in particular, the two DOE user facilities, the Joint Genome Institute and the Environmental Molecular Sciences Laboratory. The NMDC is a collaborative across 4 national labs led by Berkeley Lab and including Los Alamos, Pacific Northwest, and Oak Ridge. And we are founded on the guiding principles of data standards, data quality, data integration, and data access. So I always like to show this slide, a picture of our entire team. We're a very diverse and talented group of individuals that span computer science, microbiome research, ontologies, and bioinformatics software development. And we use our expertise and capabilities to promote all of these aspects of integration, standards, and developing a portal that links multi-omics microbiome data across these existing resources. So I encourage you to take a look at our GitHub sites for information about our metadata. You'll hear more about this from David and Sam in a few minutes. And then also the available software that we have, which you'll hear about from Ben. These are containerized bioinformatic workflows that are used in production at both the JGI and EMSL and have been made publicly available as a resource to the research community and are the foundation for which we do data processing to enable integration across our multi-omics data. As Ramana mentioned, we have this much broader vision for the NMDC beyond the pilot phase to really enable the research community to harness microbiome data. And our mission for the pilot program is to provide this gateway to fair multi-omics microbiome data for environmental researchers. And this is really why we are very interested in engaging with the Bssd science focus areas and why we're having this discussion today. So this is just a kind of pictorial view of what we hope to accomplish during the pilot phase, is to be able to take data from resources like the JGI, EMSL, NCBI, and EBI to provide the appropriate standards for describing the environmental sample information, as well as the information for how data is processed, and to enable access and search capabilities through a streamlined portal that you'll hear about later from Kirsten. Our project not only is an infrastructure project, but we have a very broad and expansive engagement component where we are very interested in developing resources to engage with research teams, scientific societies, publishers and journals, and to really enable this entire ecosystem and collaborative ecosystem going forward. As I mentioned before, standards are really critical for the NMDC, and we've spent a lot of time and effort over the past 20 months to develop community-driven standards, leveraging existing standards, as well as enhancing and developing beyond these standards within the NMDC framework. And these standards include metadata standards for environmental material, for the samples and biological samples themselves, for how these samples are handled and the data processing metadata and the bioinformatic workflows that are used to process this data. If you're curious to know more about our work in this effort, there's a great introductory about metadata and ontologies and why it's so essential to focus effort and time and using these metadata and ontology standards within your workflows and research teams. I also wanted to highlight that we are taking a very collaborative approach to developing standards and working with standards and ontologies communities. Last week, we came out with a publication that was a report from a in-person workshop that we had back in 2019. And this is really just an example of the different groups that we're collaborating with and the different inputs that we are taking and approaches in the development of the NMDC. So it's really exciting to be able to work across all of these diverse groups and to bring in all of the relevant standards for microbiome research, as well as domain-specific areas like agricultural microbiomes. So if you're interested, please read more from this publication, and we also have a lot of information on the NMDC website. All right, and with that, I would like to hand it over to David.
Thanks, Emily. So the NMDC has been developing a schema describing the entities and relationships involved in microbiome research. Chris Mungle has been leading this effort, and this provides us both a rigor of definition for all of the terms we use by tying them to existing standards and ontologies. It also forms the basis for our search capabilities in NMDC. So very quickly in this diagram, that biosample on the left actually represents a James Stegan sample taken as part of his work in the subsurface biochemical research SFA. And we can see that that's tied to both the sequencing we did at JGI and the work at EMSL here. We're showing the proteomics line here. There was also a number of organic matter characterization studies done at EMSL for those studies under using some of the same samples. So search against the attributes on that biosample, then tie all the way through to the data sets that are available that were produced here. Can you move to the next slide, please, Emily? So for those sample attributes, we're currently using the Genome Standards Consortium Mix-S standard. And in addition to using that as the basis for our attributes, we're also very involved in the development of this standard through the compliance and interoperability work group, and also through the RDF task work group. So in any meeting these days with GSC, almost half of the people there are NMDC, have NMDC collaboration or work as well. Let's move on to the next slide, Emma. GSC also makes use of the environment ontology, which Chris Mungle is involved in. This is another area where NMDC is pushing back and actually making some modifications, and we're working very hard within NMDC to make the classification of the environment easier using this type of ontology. I guess the last point I had is using Mix-S, there's actually very few required attributes. And something I think that researchers should be considering is what additional measurements you might be taking that adds value to your data sets beyond your own research. So that's something to be looking at as you're providing NMDC or the repositories with your metadata. And with that, I'm going to turn it over to Sam.
Thanks, David. So we filled out, the next slide would be great. Thanks. So we filled out the metadata schema, which can kind of, as David mentioned, be thought of as sort of a behind the scenes data scaffold. And this allows integration of other omics types beyond just genomics. An example of this is metaproteomics, pulling the functional annotation GFF file into its workflow in order to help annotate their results. which then allows users to work from the end product, such as a KO term or an EC number, all the way back up to the samples that are now known to contain those entities. Users can then allow exploration of the experimentally derived samples and the data products that are available therefrom. Next slide. In addition to building out our schema, we also wanted to build out the rest of the metadata that we had, starting with the ficus programs that we used as our initial straw man, if you will, to build out our system. As part of all of that, we've been taking an iterative approach of directly contacting the ficus PIs to obtain the important sample level metadata, which helps the NMDC fill out the initial product's data and more completely enable the efforts from slide 11. This also enabled our institutions to gain insight into which metadata are most important to the mission of NMDC. And this allows us to implement these terms into the sample intake forms. This will help lower the activation energy that's inherent in metadata entry and synthesis by getting the user community used to which terms to keep in mind as samples are collected. And finally, we'll be able to more easily enter metadata for future sample submissions. And I'd like to pass it off.
So as Emily mentioned earlier, standard is critical, and that means also all the data from different omics type, they are analyzed in a unified way so that you can compare results from different research groups. And we have packaged all the established workflows from GGI and AMSO and use all the best practice to analyze all these data. These standardized workflows are packaged in Docker containers so that they can generate similar results wherever they are deployed. All the workflows are written in the standardized WDL language so that they can run within any computing environment, and it transforms boutique bioinformatics into scalable production-grade workflows. The results from these workflows are called data products. They are also standardized, and this entire effort requires making these omic data cross-comparable and provides novel capability for microbiome researchers to search across different omics data from different groups. And all these workflows are also documented so that you can take a look into them, the workflows, and deploy them in your own environment. Next slide, Amy. So these, the metadata from the workflows are used to record them into the database so that you can perform query in the data portal, and the JSON files from the data results are also indexed there. We also provide scripts so that if you run the workflows on your own site, you can generate the same JSON standard format. The workflow documentation is online in related docs. Thanks, Emily. To provide a user-friendly environment for executing the NMDC workflows, we have provided a web interface. This web application is called NMDC Edge, and we have the web app set up in some environment accessible to all users, and it is now under internal testing. We are soon open it for beta testing, and please contact us if you are interested to get enrolled in the beta testing. All the metagenomic workflow will be available from this user-friendly interface, and all the other workflows like metatranscriptomic workflow and proteomics workflow, metabolomics workflow will be added later. And there will be separate documentation and instructions for how to use Edge and NMDC. Thank you.
I think this transitions to me now. Hi, I'm Kirsten Fagman, and I'm helping out with infrastructure on NMDC. And one of the key concepts behind the NMDC's infrastructure is that we have the capability to distribute computing and data, computing and storage, like the actual physical resources that support the infrastructure. And we can link those through a common metadata resource, or central metadata store, as we're calling it. And what this means is that when you want to use the NMDC's data portal for conducting a search, or you want to use the API to pull data out into, say, IMG or KBase or the Edge system, you're accessing this centralized resource that has all of the metadata aggregated together, as well as the data set and file locations. But the data that you access might actually live on a different physical resource. So for the Department of Energy node within this network, we're leveraging hardware and infrastructure both at EMSL, Pacific Northwest National Laboratory, as well as NERSC, the National Energy Research Scientific Computing Facility located at Berkeley Lab. Next slide. And our portal infrastructure is backed by a lot of open source software. So we have Postgres for the database. The front end is, supported by Flask and written in Vue, and all of these are containerized applications that are run in the Spin environment at NERSC. So the Spin environment is set up and meant to be scalable, so that if there's more demand for accessing the NMDC data portal, then additional containers spin up to support all of the requests coming from the community. So this is something that we're exploring so that we can make sure that we have a highly available, highly reliable resource. Next slide. And this is what I was mentioning earlier about this initial DOE node is actually backed by hardware and infrastructure that sits at NERSC and at EMSL. So what this looks like is you would have a request coming from the API or from the data portal browser. If that request is for data that's been generated at Berkeley Lab that lives at NERSC, then the browser download or the API command actually, connects to the service that lives at NERSC, pulls the data back, and the download's initiated through the browser. And the same thing happens through EMSL, where the request is made to the EMSL resources, and then the data is pulled back through the browser or copied locally through the API. And so we don't need to have all of the data sitting on the same physical infrastructure for it to be discoverable. And I think another key point is that if you would like the search to be performant and responsive, it is nice to have all of the metadata located in a central resource. And so that's part of this kind of distributed concept of having data and computing that might not be all co-located with the central metadata server, but we can still be responsive to queries to search the data as well as the access of the data. Next slide. I think I'm handing it off here to Pajal.
Hi, thanks, Kirsten. Yeah, and so we are using a community-centered design process throughout building of this NMDC data portal. And so as we're going to demo next, you'll see that what we're sharing is, one iteration of it, and we're going to continue engaging with the community and researchers like yourself to refine and build upon the features that we already have. So essentially, this is an iterative process, and we're inviting all of the researchers in the community to work with us as we improve and build this out so that it's most appropriate for your needs. Next slide. And now I'm actually going to switch over and do a quick demo of the portal. All right, is that coming through okay? Okay, great. All right, so really excited to announce that our portal is actually launched and live. So if you navigate over to data.microbiomedata.org, you can actually poke around and search for data yourself. The first thing I do want to point out is in this top right-hand corner here, you'll see my name's listed. I'm actually logged into the system right now. You do not need to be logged in if you just want to search and explore, but if you want to download any data, you do need to log in with your ORCID ID. So just pointing that out, but I'm going to just highlight a couple of interesting things here. So the left-hand side, we have various ways you can search for data. You can search based on the different sample environments using the GOLD ecosystem classification or an ENVO, and various other terms that are part of the MixS environmental packages as well. You'll also see that you can filter by the PI name, or if you're only interested in certain types of omics types of data, you can also do that. And what I actually really like to highlight here too is that we have this nifty little slider tool that allows you to, subset by a particular date if you only want to get samples that fall within that. And you'll see that all of the figures and the features update accordingly, so you can actually get a real-time update of what's available. So you can also go ahead and all of these figures here are interactive. And so if you only wanted to look for metagenomes, you can click on that and it actually lists only metagenomes and what's available. These are actually samples, so it shows you everything that's available for that particular sample. And I'm actually going to just clear this out so that I can quickly show one of our more exciting features. So if you are particularly interested in some kind of enzyme, let's just say, for example, I picked one out here. It was a butyl dehydrogenase, and this is a KEGG orthology KO term. You can actually search for that across all of the data that's in the NMDC portal. So it's really exciting because the searches across the different studies and samples, but also across the different omic types. So if this was actually, this KO term was a hit in any of that data, then it would actually return. So you'll see that this dehydrogenase was actually apparent and detected in a variety of metagenomes, metaproteomes, and metatranscriptomes. And it was found in all of the 3 data sets that we actually have here. Let's say, for example, you're only interested in, let's see, soil data. You can actually further filter that down. And it got rid of one of the studies because it was not with soil. And so just highlighting the different ways that you could search and filter. Now, if you wanted to actually go and let's say this is what you exactly what you were looking for, you can actually click on the metagenome here and you'll see all of the data products that got generated with the metagenomic pipeline here. Right now, we have a capability of going in and actually individually downloading these files. And as I mentioned before, with this iterative development, this is just our first release. And so it's not going to be final and the final product. We definitely invite you all to get involved and provide feedback and in terms of, how do you want to download your data and different ways that you want to interact with the UI here. But this is, again, I mentioned already, this is up and running, so feel free to go to data.microbiomedata.org to start exploring on your own after this webinar. That's all I have. Thank you.
Okay, I think it's my turn. I was like mid answering a question in the doc, which is great. So thanks everybody for putting the questions in there. Maybe somebody can take over and I will share my screen here. Okay. All right, can I get a thumbs up that that looks okay to everyone?
Looks great, Alicia, thanks.
Thanks. Okay, so we gave you a little bit of a background on where we came from, where some of the projects that have gone into the back end of some of this, including the metadata schemas, some of the workflows, how we're working through search and annotation and download. And then thank you to Pajao for giving a nice kind of demonstration of the portal, which just got released last week. So we definitely welcome people to go and poke around and see what you can find and let us know. Again, this is just a 1.0 release. So we are taking active feedback, and someone asked how to become a beta tester. That's great. Please send us an email to sign up. We'd love to have your feedback as we continue to do the iterative design for all of this. So I'm going to talk a little bit about kind of what the NMDC is looking for, how to partner with us, and some next steps moving forward after this short webinar. So really what the NMDC is looking for is really nice, clean, standardized metadata. Again, we're working based on using the environmental ontology, on using the Mixes environmental packages. And one of the things that we really want to make sure that we do is have really nice curated sample metadata in the system to enable the search features that Peugeot demonstrated. We're also working across multi-omics data sets, so metagenomics, metatranscriptomics, metaproteomics, and metabolomics. At the moment, we are pulling metagenomics and metatranscriptomics data from the Joint Genome Institute and metaproteomics and metabolomics data from EMSL. We are getting really close to actually taking raw sequence data from outside of the Joint Genome Institute, specifically looking at the NCBI SRA that happen to have Mixes sample metadata on the side of that. So we are starting to expand out in terms of incorporating other raw metagenomics data from other sources as well. And then I'll talk a little bit about kind of what we're trying to do in order to make the data publicly available. So we are, any data that's hosted by the NMDC is going to be under a Creative Commons Attribution 4 license, which makes it freely available for use with appropriate credit and attribution. And I'll talk a little bit how we're trying to make sure that that credit and attribution gets tracked alongside the data sets through membership that we have with ORCID, and then of course, close collaboration with Bssd. So some of the benefits of partnering with the NMDC, really we're going to continue these sorts of in-person or virtual webinars, and then eventually in person again. I think everybody's looking forward to that. But we're hoping to work with you, each one of the SFAs individually, to really make sure that the sample metadata that you're collecting is going to be NMDC compliant. And that's something that we can set up on an individual basis with each of the SFAs to really understand what kind of samples you're collecting, how it maps to the Mixes spreadsheets, what the ontology terms might be that are most appropriate, et cetera, to make sure that that standardized metadata is really nice and clean. One of the other benefits about partnering with the NMDC is the standardized processing of raw data products to produce provenance data that is really kind of unique in its ability to enable cross-data comparisons without a lot of intense collaborative discussions that generally have to happen in projects in order to do that. And so what the NMDC is trying to do is actually start to enable some of these cross-study comparisons across a variety of BER-funded projects and DOE-funded projects that aren't necessarily currently available. And then finally, really working with the community and with several of the other components of the credit and attribution system, including funders, publishers, societies, et cetera, to ensure that credit and attribution is actually tracked for all contributions to the NMDC data portal. And I'll talk a little bit more about that a little bit later. So the NMDC is really about making data discoverable. So taking those multi-omics data sets, matching them with the sample metadata and making them publicly available. We ran a survey at the end of 2020 and the beginning of 2021. It ran for about 2 months. And we got 773 responses back from the microbiome community. We asked, how do they like to find their data? And it pretty much broke out as most people are looking for environment. So that's where this standardized sample metadata becomes really important. They're looking by omic type. They're looking for a specific type of omics data that they're working for. Maybe they want to know specific annotations. Maybe they have a data product type that they're interested in. Potential sequence celerity, they have a sequence of interest and they want to find other things in the database. Where the sample is located, and then obviously what project or who collected it. And so the NMDC right now actually enables almost all of these, and we do have a project coming online soon that can look for sequence similarity. So I think the NMDC's, the UI portal for that really I think is going to be a great resource for people that are just trying to find data in a way that is interoperable and accessible. Making data comparable. Again, one of the core tenets of the NMDC is actually to take the raw data and process it through standardized workflows. which allows people to sort of select basically the data that they're looking for based on sample metadata. So again, all of those features that we talked about before, for example. This allows you to explore the data products that are produced by these consistent standardized workflows. And it allows you to compare across multi-omics data sets with confidence that the sample origin and the analyses have been curated and are documented with full provenance records. And so the additional power of this is really not just in making the data discoverable, but also making sure that you are comfortable with the fact that the data products from multi-omics data came from original sample, and we know where those samples came from and how that data has been processed. So again, making sure that you have confidence that the data are actually comparable. Making data matter. This is the really interesting challenge, I think, right now, is this concept of data citations. We are all very, very comfortable with what happens when you publish a paper and the citation records that happen with that. But it's kind of still the Wild West when it comes to data citations. And this is something that a lot of BER user facilities and funded projects are working really hard to understand how data citations move through the landscape of publications. So one of the things the National Microbiome Data Collaborative is doing is really standing up these project landing pages where we actually recommend a data citation framework that can go into a publication. So for example, here's a project landing page from James Deacon's Ficus datasets. We have a prescripted data citation format that is modeled on ESIP's formatting for how they recommend data citations. These data citations have DOIs, which are permanent identifiers for the data, and it allows people to come back to that particular project and know exactly what data they're associated with their publication. One of the things that we want to work with the community, especially all of the SFA members on this call, is really to understand your data citation needs. We want to know what goes on your project landing page. How do we represent very large collaborations like Enigma, for example, that has a huge number of investigators and organizations involved with it? How do we issue DOIs to appropriate citation pages so that you get to the granularity that you need? Each project tends to be a bit different in how they want to track this, but that's something that we are working again across JGI, EMSL, KBase, and ESS Dive to make sure that we have some best practices that we lay out for our community and how we issue DOIs and how we track those data citations, and then really working with organizations like ORCID and DataCite and Crossref to understand how we can actually provide use metrics back to the community. One of the things that we are very interested in is like if we can actually give you numbers on how many people have viewed your data set, how many times it's been downloaded, how many times your data set has been recombined into a meta-analysis and then cited in a subsequent publication. Really trying to understand what some of the incentives might be for some of these data set citations that would enable you to feel comfortable sharing your data, maybe even before you get all of your publications out of it that you had proposed in your proposal. So we look forward to really going through this process with you. We need your feedback, we need your use cases, and we really would value your input on figuring out how to do this data citation correctly. And I will say that this is not a solved problem. Having lots of conversations with OSDI and ORCID and DataCite, everyone is trying to figure out how to do this well. And I think the Department of Energy is uniquely positioned to actually have a huge role in how data citations move forward. The NMDC data use policy is available on our website. Every data set and project in the NMDC is attributed to an NMDC collaborator who openly chose to share their data with the global microbiome community. The data use policy comes in several parts. The first part has guidelines for how to actually cite data from the NMDC. For example, like on the last page for James Deacon, there was a citation for how to actually cite his project if you use data specifically from that project. We're also planning to pilot some ideas on how to combine data sets across projects. For example, if you took data sets from several SFAs that all happened to collect data around some event that happened, a big rainstorm, for example, and you wanted to combine those data sets for a data analysis, we would produce a data landing page for your particular study. That data landing page would get what's called a collection DOI in sort of the OSDI culture. And all of the collection DOI would then have DOI citations back to the combination of data sets that you used. That data set landing page with that collection DOI can then be put into a publication, and that publication then would make sure that all of the collection data that you used also gets tracked with the metadata for that publication. We also have a section on the data use policy about NMDC data contributions. That's where we state that all data in the NMDC adhere to the Creative Commons Attribution 4 license, which is free for use with appropriate attribution. And again, that attribution comes with this concept of appropriate data citation. Again, we need your feedback, we need your input, and we need your use cases to make sure that this data use policy is appropriate and it works for everyone. So a few ways to get involved. Please check out the website. Obviously, explore the data portal. We have fairly extensive documentation that can be linked either through the website or through the data portal. We welcome your feedback if you have any questions, if any things that aren't clear. Again, this is kind of an iterative design process, and we really value all of the input from our community. So if you find something that you're not sure about, or if you want to just reach out to us and ask for potential features or to get a timeline on when things might come online, please just let us know. Another way to get involved, we ask that each SFA nominate at least one representative as the NMDC point of contact. There is a table at the bottom of that shared notes document. Please put in a name and an email, and then what available multi-omics data you might be producing, and then an expected data that might be generated, for example. One of the follow-ups that we'll do with individual SFAs is that we'll start to build out these project landing pages for you to actually give us feedback on. Again, how do we make sure that we do appropriate credit and attribution on these projects that are very complicated and have lots of people involved? So that person, the name and the email that you're going to put into that document would be our point of contact, where we would actually start to connect with them. They may or may not be the only person that we need to talk to, but they would at least be a point of contact. So we have an entry point for someone. It's kind of our NMDC representative at each SFA. And then another follow-up after this call, obviously, would be to start to work with each SFA in these sort of mini sample metadata curation jamborees. Obviously, they'll be virtual. We'll come together. We ask you to bring current sample metadata that you've already collected, likely in an Excel spreadsheet, for example. And we'll walk through each one of the mandatory terms that we require for those sample metadata to meet NMDC compliance. And we're happy to do that on a case-by-case basis. Again, the NixS templates have a variety of different environments, et cetera. And so we're happy to work with you to understand which template might be most appropriate, what kind of data you might have. And then again, as David mentioned, if there are additional features that may not be captured that you think are relevant and important for us to make sure that we index, we can work with you during those follow-up webinars as well. And then finally, another way to get involved is actually to become an NMDC champion. On our website, there is an application form. It's a fairly simple, straightforward application form. If anyone's interested, Ulash is on this call. He's one of our NMDC champions. And everybody that is an NMDC champion, the first 100 gets his nice NMDC hoodies. So if you're interested in one of those. We're also going to be opening an ambassador program, an NMDC ambassador program. Nominations for that will actually open up next month, so please stay tuned for that. If you're interested in that, make sure you sign up for the newsletter and contact one of us. We'll be sending out more information specifically to this group, so everyone on this call will definitely get a notification for the ambassador program when it opens, and I'll talk about that in a minute. An NMDC champion, the basic qualification for that is it's someone that advocates for fair microbiome data. So findable, accessible, interoperable, and reusable microbiome data. And again, if you have questions about being an NMDC champion, Ulash is a good person to talk to. Our champions are also our first port of call for beta testing. So that's another way to get in to be a beta tester. So if you're both an advocate for fair microbiome data and interested in beta testing, then you're likely qualified to be an NMDC champion. The ambassadors are kind of a bit more than that. What the ambassador program actually is going to do is really start to reach out to other communities and enable them to be part of the NMDC. So the goals of the ambassador program are to empower others to share the NMDC vision within their community. And one of our goals is really to enable the ambassadors to just be kind of our local representative to plan, design, and lead community events that align with the NMDC mission. One of the reasons we're doing these is because we feel that these ambassador-led events allow the NMDC to actually tailor activities towards specific communities. Maybe it's early career, maybe there's a DEI component to it, maybe there's a soil community and an atmospheric microbiome community, and they all have specific needs that I think aren't necessarily best represented by the DOE funded team, but are actually best represented by members of the community. The ambassador program will be a year-long program. It'll start over the summer. We'll have training that will go into some of that on how to do community engagement, how to run sample jamborees, for example. And we'll also be partnering with existing ambassador programs, both at ASM and ASAP Bio, and we've also started to talk to ISME as well. So just a quick closing slide, a little refresher on what the NMDC is trying to do, really working to build out a collaborative ecosystem. We've got beta tested versions of running the workflows in Edge, for example. We're collaborating with the JDI's IMG for read-based analysis. And we're continuing to build out collaborations with ESS Dive, with Neon. Many of you might have just been on the FICUS Neon call. We're going to be working closely with them. And then putting the NMDC workflows in KBase as well. Again, how you can be involved is work with us to make sure that your sample metadata is compliant, work with us to get your data loaded into the system, set up your project landing pages, and then making sure that all of the data that's processed through the bioinformatic workflows is available to the community. And with that, I think I'll stop sharing and we can open it up to any questions that might be in the Q&A or in the document.
Great. Thanks so much, everybody. So I'm going to stop the recording now.